Hey guys, Craig here again with another installment in anatomy. This is nervous system number 2, flowing on from the nervous system number 1 video. In that first video, you remember we looked at the functions of the nervous system. We looked at the basic process of nervous function from sensory reception through to action. And we finished up on this slide here looking at a generalized version of a neuron and the different parts, and we had labeled the different parts of the generalized neuron on our mock question sheet. Again, for this presentation, you'll want to have your mock question sheet handy. That is this bad boy here called 22266 Anatomy Physiology Test 1 Mock Questions here. And the last thing we've done here was under question 17, we've drawn a generalized neuron and labeled the parts. Today we'll continue and finish off these mock questions so that you have everything you need for section 1 of your test, the nervous system. So let's dive into it. So in this first part here, we're going to look at each part of this neuron and find out what each of these parts we labelled at the end of the last session do. We'll start at this end up here with the dendrites. So just to familiarise yourself with this guy again, imagine that this is a sensory neuron, that these nerve endings here we see as dendrites would be plugging into somewhere like the skin, and they would have receptors on their very ends here, and those receptors would be picking up things like pain or pressure, heat, temperature, those kind of things, and relaying that information to the next part of the nervous system chain. So if this was a sensory neuron, that information would get sent via electrical impulses down into the soma here, then shoot along the axon and right to the ends where the synaptic knobs here would meet another neuron. In this case, if this was a sensory neuron, the next one would be an interneuron within the spinal cord, whose dendrites would come up and meet each of these little points here, and we'll see some pictures that show that soon. So let's have a look at those different bits real quick. The dendrites, as I said, is where the sensory receptors are, and basically they receive the signals and then pass on that information in the form of electricity. The more dendrites that a neuron has, so the more of these little antlers, the more information it can pick up, and the better the feedback that the central nervous system gets about what's going on. Those little bits of information, those little impulses, are sent down the dendrites, the little antlers there, and congregate in the central head of the generalized neuron. In different neurons, the soma is actually going to be in different places. Sometimes it's halfway along the neuron, and other times it's at the head, like this one is here. In the soma, or cell body, you have the nucleus, and in the nucleus is where you find the DNA information for how to make or repair a cell, such as this one here. So there is the soma, would make up this head on this occasion, and there is the nucleus. The axon is the tail of the neuron, and it's effectively the cord that the electricity runs through to get from point A to point B. So it will carry the messages, in this case, let's say from the skin all the way to the spinal cord and the central nervous system. So in some cases, for example, from the toe all the way to the base of the spinal cord, that could be more than 1 metre in length, i.e. the entire length of your leg. So its main role is to just conduct these electrical impulses to the next point of the chain. So this is the axon here, underneath these yellow blobs going all the way to the end point. Now along the length of the axon, you're going to find those little yellow blobs, and we'll just go back and have a look at them quickly. These guys here, known as the myelin sheaths, these are effectively fat layers, so layers of insulation that are wrapped around the axon, and the fat acts as an insulator and a conductor, so it allows electricity to be funneled quickly, more quickly than if it was unmyelinated. So as we learn through our life cycle, we lay down these layers of myelin sheath, and that helps us conduct information quicker around the nervous system. So as you groove a new task or learn a new thing, you'll myelinate that axon, and therefore the messages will move more swiftly, allowing you to act more swiftly and receive quicker information about what's going on around the body or outside the body. Now these fatty layers are laid down by the Schwann cell, which you see here, the orange dot. The Schwann cell effectively is the thing that produces these fatty layers and maintains them. So you could have as many as 100 layers, as the notes say here, around the axon, forming that myelin sheath. And the whole point of this nerve conduction is to get it to the end point, known as the synaptic knob, or what we'll learn as the presynaptic terminal. And what presynaptic terminal indicates is that it's one side of a meeting point between two nerves. Okay, so this is the pre side. There's then going to be a little gap, and then there's the post-synaptic terminal, which you'll pass the information over to so it can continue its journey. So in the picture to the side here, what we see is a sensory neuron coming in and attaching into the spinal cord. So the next point of the chain would be, if these were the synaptic knobs that I'm pointing to right here, then we would have a meeting point with another neuron within the spinal cord that would then take the information up to the brain. And it's that meeting point, that junction of meeting between two different nerves that we're going to move into next. So here we can see two nerves meeting. We see the red neuron here. Let's say that was the sensory neuron. Here's the axon branching into its synaptic knobs, and you can see that the synaptic knobs of the red neuron, the sensory neuron, are meeting with the dendrites of the blue neuron, which in this case could be an interneuron. So we're forming that connection between the two neurons to pass on the information. Now, these connection points can have multiple times throughout one nervous system activation. In this case, if it was sensory neuron passing over to interneuron, that message would now, when it gets into the blue neuron here, be inside the CNS, i.e. inside the spinal cord. It may then travel up to the brain and actually be passed over to multiple other interneurons. So the same process would occur, and it would bounce around the brain to the different areas it needs to before a decision is made on what to do with that information. That information would then be relayed back down an interneuron, and then finally, if this was an interneuron now, passing out of the spinal cord would pass it on to a motor neuron, and the motor neuron would effectively pass the message on to the thing in the body that can do something about it. So the final point of the chain would not look like this. It would be a motor neuron with its synaptic knobs plugging into a muscle, plugging into an organ, or plugging into a gland that would cause an action to occur in the body. So the meeting point, and we've circled one of them here, this little yellow circle here indicates one meeting point between two nerves here, and this guy we're going to know as a synapse. So the synapse is a junctional point of contact between two nerve cells, allowing that nerve to communicate. And as you can see in this picture, as we go back, there are multiple synapses between each neuron. You can see multiple dendrites meeting with multiple synaptic knobs there. The essential components of one synapse are the arriving synaptic knob, which is the presynaptic terminal. There's a minute gap between the two meeting points, known as the synaptic cleft, and then the dendrite on the other side, which is the postsynaptic membrane, in this case a dendrite of another neuron. So here we see one of those meeting points up close. Here is the presynaptic terminal or neuron. Here is the postsynaptic neuron. And here in the middle, this tiny little gap here is the synaptic cleft. So with that in mind, let's just solidify that information by going back into our mock questions for a second here. And we'll go to question 12 here, define a synapse. And we can define a synapse as the meeting point between two neurons, allowing them to communicate. That's the meeting point between two neurons, allowing them to communicate. So feel free to pause or go back in the video to get that definition down. Question 13, where would you find a synaptic cleft? You'd find it between the presynaptic terminal and the postsynaptic membrane. Now you'll see these referred to in different terms a couple of times through this. There are always different names for things in anatomy and physiology. Maybe the simplest way to remember it would be using the term neuron. So presynaptic neuron, postsynaptic neuron is fine. Probably more specifically known as presynaptic terminal and postsynaptic membrane. And you would find the synaptic cleft as the gap between those two. Now, what's the relevance of that gap? That's the next question. So let's just pop back into here. So why have a gap at all? The answer to that's not easy. We don't know why there's a gap there. Why don't they just plug directly into each other? Why don't they just pass the same electricity through both neurons? We don't know the answer, but what we do know is how the electricity is conducted across the gap. Now, the first thing to note is that electricity or impulses can't cross the gap directly. So what needs to happen is the electricity that arrives down this synaptic, presynaptic terminal here, then needs to stimulate a response that will cause a new impulse to be conducted on the other side. Okay, so we have these little guys in here in these little white circles here. These are called vesicles, and these are called neurotransmitters, these little black dots in here. And neurotransmitters are effectively chemical couriers in the body that pass messages on. They're stimulated by the arrival of the impulse here. So in this case, we will use the term impulse a lot, but we'll also use a term called an action potential. And an action potential is some electricity that is arriving that has the potential to cause an action. In this case, the action potential causes the release of neurotransmitters across the synaptic cleft here. So here you can see the little neurotransmitters crossing across. And here you'll see these little pink channels here. Now these are the ion channels, and they have little receptors on the end. You can see some of these little dots here attaching to the ends, and what that will cause to happen is it will cause the opening of these channels. So the two pink cylinders there will slide away from each other, leaving a gap in the middle. Now the opening of those channels is really important. They're called ion channels, and ion indicates that there's a positively charged substance that is somewhere around that wants to come in those channels. And in this case, the entire nervous system is bathed in extracellular fluid. And extracellular fluid includes positively charged ions like sodium. And there are more, but we're just going to use sodium as an example here. So if you just imagine this whole thing here, the whole nervous system, the axons, the dendrites and everything being bathed in this extracellular fluid, that means there's abundance of positively charged substances, i.e. sodium, floating around in the space. When the neurotransmitters open the channels, those positively charged ions of sodium come pouring in through the channel. And what that does is create a new positive charge of electricity on the other side here. Now, if enough neurotransmitters are stimulated across the gap and the channels are opened up for long enough, then enough positively charged energy comes in to cause the neuron on the other side to reach its threshold, and therefore the nerve will fire and pass on the message to the next point of the chain. So let's explore that a little more. So the presynaptic terminal forms the end of one axon, we know. In this case, it could be a sensory neuron, and it contains the neurotransmitters. And those neurotransmitters are the things that will cross the synaptic cleft to pass on the message to the other side, which in this case would be the postsynaptic membrane. The minute space between the pre- and postsynaptic terminals is of course called the synaptic cleft, and we know that electricity can't cross. Only the neurotransmitters can cross. When the neurotransmitters cross, we can cause a new impulse to generate on the other side in the postsynaptic membrane, and that happens of course by binding to the receptors on the postsynaptic membrane, opening up those ion channels and allowing positively charged ions to flow through. So on the next slide is a video, and I'm not going to show you the video on this video lecture here, but you can access it through the full Prezi that's found above this video, nervous system Prezi. And what you'll see on that in the first sort of minute, and I suggest you only watch the first minute of that because the rest of it won't make a lot of sense to you at this point. But what you'll see on that video is the process of the action potential arriving at the synapse. You'll see color-coded neurotransmitters making their way across. You'll see them binding to receptors and opening up different channels on the other side, allowing positively charged ions to flow in. So what you'll see on the next part of the video is a soma, or cell body with its dendrites sticking out. And you'll see that it's bathed in fluid, and that fluid will be represented by some space outside and some yellow dots. And those yellow dots are sodium. So what you'll see is the channels will open and sodium will make its way into the soma. And when that happens, you'll see a white light starting to grow in the middle of the soma, and that's indicating that this positively charged sodium is causing a new charge, which is building up. And the building up is really important because in order for the neuron to pass on the message, enough positively charged sodium has to flow in to raise the electrical stimuli inside that soma to the neuron's threshold, which we'll talk about soon. But at this point, just to keep things basic, each neuron has its predetermined threshold, and if enough electricity arrives to reach that threshold, then that neuron will fire and pass the message on. So neurons don't partially fire, they either fire 100% or not at all. So we'll just skip past that video and get into, and you can watch that in your own time. And we'll move on to that process of summation very, very soon. But what I want to touch on first is just to get a brief thing out of the way, and that is the difference between these voluntary and brain-orientated decisions that go on in the human body and reflexes. So this is a nice little short piece, just to explain that there is no difference in the neurons involved in a reflex compared to a voluntary situation. So in this picture here, we're looking at a reflex, and it's a sensory neuron here plugged into the skin, as you can see, and let's use pain as a classic example of this, right? So if pain receptors in the skin were to fire, and the amount of pain was enough to get a significant amount of electrical stimuli to flow down the sensory neuron here, we would see it processed by the cell body, which in this case in the sensory neuron is actually midway down the neuron. Not to confuse things, you don't have to worry about that too much. But that information would trigger a reflex response if the pain threshold was achieved. You'll notice that it plugs into the spinal cord and that an interneuron picks it up. And in this case, the interneuron is very, very short because it's a pre-programmed reflex interneuron that says if this much pain occurs from this sensory neuron, send out this pre-programmed response. And the pre-programmed response would be flicked out immediately out through a motor neuron and plug into a muscle and get us to move our arm out of the way of the fire or the hot object that had burned it. Now that would occur without any involvement of the upper central nervous system. So the brain wouldn't be involved in that at all, and it's to save time. Let's just get it out of the way, and then a message from interneurons would continue up to the brain, and the brain would eventually, not very long after, but would pretty quickly after, know that a painful stimuli had occurred, and you would register that as pain. But you would have moved before the pain had even reached the brain and been recognized by the brain as pain. Now, the question in the exam that you'd be familiar with, it's a kind of trick question that asks you what neurons are involved in a reflex. And of course, the answer to that is all the same three that are usually involved. So what we'll do now is just dip into our mock questions again and answer a question that relates to this one here. There we go, number 18, which asks you what are the three types of neuron involved in the reflex arc? And your answer is, of course, that it is the sensory, inter and motor neurons. So chuck that answer in. We can also add one more into here, and that is question number 14, which asks us what causes the release of neurotransmitters at the presynaptic terminal? What causes those neurotransmitters to cross the gap? And the answer to that is the arrival of an action potential, or an AP, you can call it, capital AP. And action potential, just to remind you, is a piece of electricity that has arrived at the presynaptic terminal. Cool. So there's a little video on the reflex situation here that you can watch on the full Prezi. We'll just skip over that for the moment. You can have a look at that in your own time. And we'll get on to this next wee bit, which is all about the charge within the neuron that causes it to fire. So we've talked about how sodium plays an important role in this. Sodium is required to flux into the cell body to get the positive charge to occur on the other side of the synaptic cleft. And this actually needs to happen not only at the soma, but in order for the message to pass its way down the axon, we need this constant influx of sodium occurring to keep the charge going as it goes down the axon to the end. So along the entire process that a piece of electricity makes its way down the neuron, you're going to see this influx of sodium going into the nerve and keeping the message going. We'll see a wee video on that shortly. So, positively charged ions enter the nerve. This causes an increase in positive charge inside the nerve, and an increase in positive charge is known as depolarization. So depolarization simply means increasing the positive charge within a neuron, the amount of electricity that's there. Now at rest, most neurons will sit at about minus 70 millivolts. They'll be in a negative electricity state. And the main reason for that is the protein, the actual protein components that make up nerves. And protein is a negatively charged compound. You can see the negative protein molecules here. It also has some positively charged potassium inside it here, but you can see all of this positively charged sodium waiting on the outside, ready to be escorted in once the channels are opened up. So if we were to read the electrical levels of a resting neuron with no electricity flowing through it to cause any action or anything like that, then it would have a baseline of minus 70, and that's the resting membrane potential. Now, in order for a neuron to fire, we need to depolarize the neuron, which means increase the positive charge inside it. And each neuron has a threshold, and the average threshold of a neuron is about minus 55 millivolts. So what that tells you is we need to increase the charge inside that neuron, or a neuron, by about 15 millivolts from negative 70 to negative 55 in order for that neuron to fire. Now, if the number of impulses arriving doesn't cause that change to happen to 15 millivolts or above, then the neuron won't pass the message on. It's called the all or nothing principle. It will either fire if it reaches the threshold, or it won't fire. So, let's have a look at that process in action here. So this is a picture process, and this will be followed by a little video to show you in action there. And what you're seeing here is the ion channels have opened and the sodium is starting to crowd in, it's starting to pour in, and what that does is raise the polarity of the neuron. So this neuron is depolarizing. It is increasing in positive charge as these little positively charged ions come crowding in. And what we see on the reading over here is that we move from our minus 70, we start to depolarize. So we start to get an increase in terms of millivolts or electricity going through. Now this one has obviously gone way past the threshold. The threshold was minus 55. This one's achieved 0. It doesn't matter how far you go past the threshold. The same result is going to happen. The electrical field is going to continue down the axon until it reaches the end. The neuron will fire. Now this little video here, I'm just going to skip over it because the quality of video when we play it on these video lectures doesn't usually come out very well. But I encourage you to go to the full Prezi and have a look at this video. It's going to open up and start with messages being passed into neurons within the brain, and it's going to dive into the axon of 1, and it will show you positively charged ions represented by red pluses, meaning sodium, diving in and diving out of the axon as the electricity goes through. It'll also show you the action that occurs at the synaptic cleft, so where the presynaptic terminal meets the postsynaptic. You're going to see a white charge arriving, and if you look very closely, you'll see the little red neurotransmitters in the end of the presynaptic terminal jump across the synaptic cleft, attached to the tops of the membrane ion channels on the postsynaptic membrane, and then you'll see the positively charged sodium ions dive in, little balls of electricity dive in. And you'll see also at the end of it a white light starting to strengthen on the far side of the synaptic cleft, and that's the depolarization occurring. So that's the new electrical impulse being made on the other side. All right, a key sentence on this one here, the all or none principle, you need to be aware of this. It looks a little tricky, this quote, and we will be writing down some of this quote on our mock questions in a moment, but we now know some word substitution that we can use to make this quote easier. If the depolarization, think positive charge, if the positive charge of a neuron changes its resting potential, which was negative 70 millivolts, equal to or past its threshold, which is about negative 55 millivolts, then an action potential, new piece of electricity, will be formed and the neuron will fire. So basically, if the increase in the positive charge that comes into the neuron is enough to reach the threshold, the neuron will fire. And if the charge isn't enough to reach the threshold, the neuron won't fire and the message won't be passed on. Let's take that information now and put it into our mock questions. we're going to roll down to question number 19 here. The resting membrane potential of a normal neuron is minus 70 millivolts. Don't forget the minus there, really important. What is the result if, and don't worry about this EPSP, this is basically action potential. This is the little pieces of electricity that are coming in. If the little pieces of electricity that come in build up and change that resting membrane potential higher than negative 55 millivolts, what's going to happen? And the answer to that is the neuron will fire. And finally, what does the all or none principle state in relation to a neuron's firing? It states what we had on the other slide there, and I'll just bring that back and you can fill this in, number 21, as we go back to this. Show you what to write in here. If we could write down, if the depolarization of a neuron reaches the threshold, the neuron will fire. I'll read it again. If the depolarization of a neuron reaches its threshold, then the neuron will fire. And that is essentially the all or none principle. Now, of those little pieces of electricity that arrive and congregate as the little bits of sodium come in, the positively charged pieces of sodium come in, and you imagine them travelling down the dendrite to the soma and sort of forming in the middle of that cell body, well, we can pretty much establish that one of those little pieces of electricity is not enough to create the threshold reaching that we need. So one of those little pieces of electricity is about 0.5 millivolts. We know that we need to reach 15 millivolts of increased positive charge to achieve the threshold. Therefore, we need about 30 of those little pieces of electricity at least to congregate to form a threshold, reach the threshold and fire the neuron. There are two ways in which these things add together, these little pieces of electricity. Now, the adding together of electricity to form a sum of millivolts that will reach the threshold is known as summation. And there are two types of summation. We're going to keep this really simple. If just one synapse, one meeting point between the neurons is firing, that's called temporal summation. And that's going to happen in a very low intensity type situation where there's something ticking away in the background that adds up to the threshold and causes a minor action to occur. Now, when we want a lot more information coming in, or we want to act very quickly on something, then we get far more than one synapse firing at a time. We get multiple synapses firing. That's known as spatial summation. So one synapse firing, temporal summation, and more than one synapse firing, spatial summation. We'll just take the picture here and just illustrate that. So if just one of these little guys is firing its messages across and they're making their way to the soma and adding up to the threshold and firing off, that's called temporal summation. And if multiple ones of these, maybe even all of these guys are firing at the same time, you can imagine the threshold is reached much quicker, more information is coming in, and the neuron will fire much quicker. And that's called spatial summation. We'll get back into our mock questions and just add in these two questions here. So all I want you to write under number 22 to find temporal summation, when one synapse fires to reach the threshold. And number 23, spatial summation, when several synapses fire to reach the threshold. Pretty simple. So while we're here, we probably should have done this earlier, but we'll do it now. We'll have a look at number 25. And essentially in here we need to draw a synapse. So we need to draw one presynaptic terminal and one postsynaptic membrane and leave a little gap in the middle and label these guys presynaptic, postsynaptic and synaptic cleft. And it's going to be a simple labeling task on your exam as well, where you just recognize where each of those parts are. So really easy marks there. So that's basically just drawing one of these meeting points between this guy and this guy, but obviously leaving a small gap in the middle to illustrate the synaptic cleft, which is here, but we can't see it because we've panned back too far. Okay, so the messages have got through, they've caused the desired action, and now we need to stop sending messages, and we need to let the neuron go back to its normal resting charge. So once the action potential has passed through the neuron, and we don't need to create any more, the electrical charges return back to normal as the electricity moves through. When you go back to normal, this is called repolarization. And the exact opposite chain of events occurs. What we can see now is sodium now makes its way out of the neuron where it pulsed in and then now flows out. And you saw that on that video before. The positively charged ions went like a wave. They came in and then they came out, kind of like breathing. And what happens when they come out, obviously, is that the positive charge they brought with them drops away and we go back to resting. And that is called repolarization. There is a question in the mock questions that we'll get into now. We'll just finish that off. And that is question number 24, what is the process of repolarization? And that is when the neuron returns to minus 70 millivolts. When the neuron returns to minus 70 millivolts. Done. So while we're here, now that we've drawn in our presynaptic terminal and our postsynaptic membrane and our synaptic cleft here, you just see number 26 here asks what the function of these two structures are. And that's pretty easy now. We know that the presynaptic terminal receives action potentials, so the electricity, and releases neurotransmitters. So the presynaptic neuron receives action potentials and releases neurotransmitters. If we could write that in. And then the postsynaptic membrane receives neurotransmitters and, if enough, creates a new action potential. So I'll read that again. The postsynaptic membrane receives neurotransmitters and, if enough, creates a new action potential or a new impulse to be sent along the neuron. Alright, so we're getting there. We're motoring through these mock questions that will effectively become your study sheets for the exam. There's a little video in the presentation at this point here just asking the question, can you block these messages? And you can see from the video, could be a pretty intriguing video. It's a dude getting kicked in the knackers. And what you'll find in that video is a karate sensei, effectively, who's trained himself to block these messages of pain by getting kicked in the nuts thousands of times. So, yeah, go have a look at the video and see a world record kick in the nuts and how this guy just takes it. And they talk a little bit about the processes of the nervous system that are going on there. So a lot of the learnings in this one are around the neurotransmitter called substance P, which processes pain. And what we know is that if you expose yourself to different stimuli regularly, then the way in which your brain processes that information changes. So if you become, it's kind of like going to the gym. If you go to the gym and you have your first leg workout in a couple of years, you know that that's going to leave you really sore the next few days. Okay, so you're processing a lot of that substance P, which is telling you that painful stimuli and damage has been done down there. But as your muscles strengthen and get used to those types of workouts, you don't process as much. It's not that you're not doing as much damage as before. Your brain is simply just not processing that as a negative or really bad pain. So you learn to process through the process of doing. So the more you do something, the easier your brain finds it to process that info, and the less panicked it is, I guess, about some of these situations. So I'll let you watch that one on the full prezi. So guys, at this point, we've covered quite a lot of the nervous system. We've only got a little bit to go, and that's this final part on the right-hand side here. We've covered how receptors pick up information and pass them up through the sensory division, the sensory neuron, into the brain and spinal cord for either a reflex or a voluntary or involuntary response. Then when a decision or reflex is decided upon, we send that out through a motor neuron to the motor division. We're now going to look at how we differentiate the two different types of outgoing messages to the body and how they cause action around the body. So the motor division can be split into somatic and autonomic. Okay, and the easiest way to think about those is autonomic is virtually automatic. It means things that happen without your conscious involvement. So we're talking about a lot of your internal processes here, like where your blood goes in your body, oxygen level monitoring, how often you breathe and your heart beats, etc. You don't consciously control that. It happens in the background, and that is the autonomic nervous system. Now, the autonomic nervous system also has a secondary component to it, and that is another thing that you can't control, and that is, of course, your reflexes, your pre-programmed primary reflexes, which are really revolved around a genetic predisposition to reacting to dangerous stimuli in a certain way. It's also known as the fight or flight response. So you are either more aligned to run away from danger and cower and hide, or fight, or in some cases even freeze. So lately, fight or flight has been changed to a three-pronged attack called fight, fright, or flight. And the fright is, of course, if you just freeze like a deer in the headlights. The other side of the motor division is, of course, the voluntary decisions that you make, the things that you decide to do. And the somatic nervous system is responsible for voluntary movement. So mainly movement of skeletal muscle. That causes us to be able to write, type things, catch a ball, lift a weight, and all those kind of things that we mean to do. So we see either voluntary decisions here or involuntary decisions here, and then we see the two splits of the involuntary decision. This is sympathetic or fight or flight system, and this is your rest and digest, your housekeeping type system. And we have very limited control over these guys. We can influence them by making voluntary decisions, but they will happen with or without our conscious brain involvement. So the somatic division, like I said, is the one that we use to create voluntary movements, like kicking a ball or picking up a weight, whereas our autonomic nervous system primarily is involved in those internal processes, the involuntary functions of heart rate, blood pressure, breathing, etc. So here are the two splits of that autonomic one, as I mentioned, the fight, flight, or fright response, known as sympathetic. So really important that you know the two names for the division. Sympathetic is fight, flight, or fright, and that is how you respond to stressful situations. So an example like burning your hand, someone jumping out and scaring you, that kind of stuff. Whereas the parasympathetic division is your housekeeping system, known as the rest and digest. So make sure you know rest and digest is parasympathetic, sympathetic is fight, fright, or flight. One non-PC way of remembering parasympathetic division is paralyzed. If you're paralyzed, you can basically rest and digest. There's nothing else you can really do. Alrighty, so we're on to the last bit, and that is chemoreceptors and proprioceptors. What we're going to do though is dive in and answer the questions from the stuff we just talked about and get them done. So we'll go back up here. So we're finishing off page 1 here, the different divisions. What division of the, number 9, skeletal muscle, is controlled by which division of the PNS's motor division? And the answer to that is the somatic division, so SOMATIC division. Number 10, which division of the autonomic nervous system is known as the fight, fright, or flight system? That is the sympathetic nervous system. And number 11, what division of the autonomic nervous system is known as the rest and digest system? And the answer to that is the parasympathetic nervous system. So there may be one question left. This is this little one here, number 8. This is more of a generic question, just to finish off this page here. The nervous system must work more as the movement required becomes more. The answer to that is either complex or difficult. So the more involved the decision-making process that you're trying to do, the more tough the thing you're trying to learn, then the more nervous system activation you'll need to get it done. If things are really easy, you don't have to devote much attention to them. Okay, so if you're really accustomed to like catching a rugby ball and people are throwing a ball at you, then you can talk and joke and catch it at the same time. But if it's completely new to you, you probably won't be able to hold a conversation because you'll be so focused on trying to do the task. Alright, so we'll just get in and finish this off. We are almost there. We're going to look at, finally, the receptors that are involved in sport and exercise, and particularly those that keep us from injuring ourselves. So we're just going to look at 3 different types here. So the first we're going to look at are muscle chemoreceptors. These are sensory receptors in the body that monitor the levels of chemicals that are floating around our bloodstream or within our muscle. And the other ones we're going to look at are proprioceptors, which are found in our joints, muscles, and tendons, and these are the guys that keep us safe during exercise. They allow us to do movements safely and not cause potentially harmful injuries. So chemoreceptors, Not quite what you'd think. They will monitor dangerous chemicals in the body, but actually they're there to monitor chemicals that are quite often found in the body and let you know the levels of these chemicals and what needs to change in terms of your body's internal maintenance system to keep levels of dangerous ones down and make sure we get enough of the good ones. The key chemicals that they monitor are oxygen and CO2 levels within blood and muscle. So you have constant monitoring of the oxygen levels found in your blood. And of course, when you start doing more exercise, when you're out there and you're running around, your muscles start utilizing more oxygen, and therefore you have less oxygen in your blood. Now, these receptors will monitor that, and they will tell the brain, hey, there's less oxygen in the blood, we need to get more, and then the brain will cause your diaphragm to work a little harder, and your breathing muscles to work a little bit harder, and you'll start to breathe harder and faster. Obviously, doing exercise will increase CO2 levels in your blood as well, as the CO2 is a waste product of any cellular metabolism. And of course, we're exercising, we're burning fuel, and CO2 is coming out. So then blood levels of CO2 will start rising. The brain will be informed by chemoreceptors and will start to increase your heart rate to get that CO2 back to the lungs and increase your breathing rate to get you to blow that CO2 out and match the requirement of maintenance within the tissues. So these guys basically provide your CNS, your brain and spinal cord, with information about the metabolic rate of your muscle activity. And what that means is we know that when you're working out, the levels of CO2 and O2 that are found in your bloodstream are an indicator of the fuels that you're using and the metabolic rate that you're achieving. We know that fat requires oxygen to combust, and that's why we use it mainly in the aerobics energy system, because of course you have a constant supply of oxygen that's meeting the demands of the muscles. So we're able to utilize fat as a fuel source. So we know how much oxygen's in the air, we know how much is coming in, 21% of the air is oxygen, and then we measure the amount of oxygen coming out, and that tells us how much oxygen you're using during exercise, and that's an indicator of your fuel use or metabolic rate. And in this case, if we were utilizing large amounts of oxygen, we'd be burning more fat. We can also measure your CO2 levels coming out, because we know there is very little CO2 in the air that you breathe in. When the CO2 levels rise dramatically and you're breathing out during exercise, we know that you're mainly burning carbs, which are not a very clean and efficient fuel source and produces a lot of CO2 waste. And therefore we can have an indication of what fuels your muscles are using. What you need to write down in your mock question, however, is just a simple sentence. If we go down to these guys, you'll find them down here. Number 16, what's the role of chemoreceptors? They monitor the metabolic rate of muscle activity. I'll say it again. They monitor the metabolic rate of muscle activity. And then finally, we have the proprioceptors. The first of these is your joint kinesthetic receptors. You can guess where you find them. In your joints, it's in the name. I call them JKRs because it's quite a mouthful. And you'll remember from our studies in anatomy that synovial joints have joint capsules. They have a capsule that encapsulates the whole joint, and it's filled with synovial fluid. Now, these joint kinesthetic receptors reside in the joint capsule, and they're very, very useful to us. They are effectively our feel, what we experience as feel. So they tell us where our body parts are, they tell us where our joints are positioned, where our body parts are positioned, and they give us constant feedback about that, so we don't have to look at our body parts to know where they are. So if you were to close your eyes now and extend one arm out to the side, if you were to bend your elbow and touch yourself on the nose with your finger, I'm pretty confident you were close, if not bang on. Now, how do you know where your nose is? How do you know what the distance is to there? You know this because of your joint kinesthetic receptors, which told you where your arm was positioned, which told you where your nose was positioned, and told you how to get there in a seamless sort of movement without even looking. So the benefit of this is obviously you don't have to watch everything all the time. If you were running and trying to track a ball in the air and not looking at your feet, if you didn't have these guys, you wouldn't know where your feet are and you would effectively trip over. So they allow you to focus on other things by giving you an intrinsic feel for where your body is at all times. Now, when you injure yourself, these guys, particularly a sprain, like a joint injury, these guys are injured also, and their effectiveness lessens. And that's why it's really important to do your proprioceptive activities and rehab after a sprain, and that's the type of stuff we do with wobble boards and balance and single leg movements for a sprained ankle. And if you're like me, you do them a few times and then you strap them up and then you get back into playing. But that's a recipe for disaster, really, because you need them to heal properly so that they can give good feedback. Otherwise, you're getting not incorrect feedback, but incomplete feedback, and it means you're more likely to turn your ankle again because you don't know exactly how it's sitting in space. So balance is a great example of JKR's working. All of the minute adjustments that you make when you're trying to balance on one leg or on a high wire, as this guy's doing, are unconscious and reflex-orientated messages that are being generated by joint kinesthetic receptor impulses. So they're coming in and telling you what's happening, and then your body is adjusting to that subconsciously. So, as we overbalance to one side, say on one leg, then messages will come from the lateral ligaments and capsule of your knee saying we're going too far to the outside, and you'll get a reflex response by contracting the vastus medialis on the inside of the knee, and that'll pull you back into alignment. And because you're in an unbalanced state, you'll probably go too far to the inside. And then your medial ligaments and medial capsule send sensory information saying, no, go back the other way. And that's why you have this constant sort of overcorrection and wobble at the knee as you balance. All right, second last one we look at is muscle spindles. Muscle spindles, as you can imagine by the name, are found in your muscles, and they monitor static and dynamic muscle length, which means they monitor your muscles length when you're sitting or standing and not moving, and they also monitor it when you are moving and running and jumping and doing all those things. The idea is that they will protect you from tearing a muscle. Now, most muscle tears occur in the extension phase of muscle, where you sort of violently extend and then try to contract from the point of maximal extension, and that's generally where the muscle will tear. So what they do for you is they sense if a muscle is being overstretched, and if they think it is being overstretched, they cause that muscle to contract to save itself. Okay, so that prevents the tear from happening. So a great example of that, I'll just show you where they reside. So here you can see the muscle spindle. It's in between the muscle fibers here, and here's a sensory neuron sending off that information about stretch. It will have a paired motor neuron that comes down and fires the muscle to contract, to save itself if it believes it's being overstretched. So there you go, you see it in its sheath there. These are muscle fibers with their motor neurons attached, and this is a sensory muscle spindle, which is in its only sheath and is just there to sense extension. Now, a great example of this is if you're doing a hamstring stretch, you're feeling a bit tight, and you put your heel up on a table, straight leg, and you lean forward, and you'll get so far before you get that sense of kind of the muscle fighting against itself. So that's how a stretch actually occurs. You start to stretch the tissue, the muscle spindle gets involved, thinking that you're going too far, and causes the hamstring to contract on itself, and that's where you feel the fight occurring within your hamstring. One, you're stretching voluntarily, and two, it's fighting back and contracting involuntarily. Now what you usually find is if you try to force it and lean forward too far with a straight leg, your knee will bend, and that's where the muscle spindles decided, no, that's too much stretch, and I'm going to bend the knee, which is of course contracting the hamstring to bend the knee, and cause the release of tension. So that's essentially going to save the muscle from being taken too far. But what we also know about stretching is if you were to hold that for a period of time, that stretch, and then release it, and then go back again, you can often go further than you could the first time. Now that indicates that muscle spindles can habituate to a new length, meaning they have a preset or predetermined length that they determine is too far for your muscle to stretch without tearing. But if you were to hold a stretch and cause a flexibility change within the muscle, typically over 15 to 30 seconds of holding, and then go back and stretch again, you'll be able to go further because the muscle spindle will go, okay, he obviously wants to do this. This is a voluntary stretch that's been performed here. Maybe I can allow that to go a little further, and you'll be able to go a little further. But of course, if you try to go too far, the muscle spindle will kick in again, and the stretch will end as the knee bends. Another classic example of the muscle spindle, so there's the hamstring thing that we're talking about there. The actual stretch you feel is, of course, the muscle fighting back against itself. The question then is, why hold the stretch? Because holding the stretch for 15 to 30 seconds helps the muscle spindle habituate or reset, so that repeating the stretch should result in the ability to stretch further, and that's how you increase flexibility. As a final example, you guys are probably familiar with the patellar tendon reflex test, where you hit someone on the knee with a hammer. You can see over here getting hit on the knee with a hammer, and we know that the result of that is usually a pretty violent sort of extension of the knee. So what's happening there? That is your muscle spindle kicking in. So what happens is we're hitting the patellar tendon just below the patella, which is going to pull the patella pretty violently down as we hit into that space. The patellar tendon at the top here we know is attached to your rectus femoris, and the end fibers, the distal fibers of your rectus femoris, are going to be stretched pretty quickly and violently. That's going to fire the muscle spindle, so we can see here the muscle spindle sends its sensory information up. We get a reflex interneuron response, spits it out through a motor neuron to the rectus femoris, which causes the rectus femoris to contract violently, and of course that kicks your knee up in the air. And we should expect that to happen quickly because it is a reflex, and that shows you that the nervous system is working the way it should, that those muscle spindles are firing. If they weren't, then you would be at danger of muscle tears, because of course if it was a very slow response, the muscle would let you go too far and the muscle would tear before the spindle kicked in. So the final one we talk about is the Golgi tendon organ. Again, in the name, it's monitoring a tendon, and this time it's monitoring tendon stress, so the amount of force that's being put through the tendon. And if it believes that the tendon has too much force going through it and might snap, it will switch off the contracting muscle that's causing the tension. So here you can see muscle fibers culminating in tendon or collagen fibers into one strip of tendon, and here's the Golgi tendon organ that's there monitoring that tension. So tension increases in a tendon when muscle fibers contract forcibly. So you're lifting a weight at this point. You're doing a bicep curl and the bicep tendon is really getting loaded with force as the bicep muscle pulls on the forearm through the tendon. Okay, so they're found at the musculotendinous junction, which is where the muscle turns into tendon, and they detect tension applied to the tendon by the muscle. Okay, now if excessive tension occurs, they will inhibit the contracting muscle. That's the key thing I want you to remember. They will switch off the contracting muscle, which will end the tension on the tendon. And of course, if you're lifting a weight at that point, you're going to fail. So your muscle will switch off and you'll drop the weight. They can also cause the antagonist to contract, so the muscle on the other side. So in the case of the bicep curl, they could cause the triceps to kick in, which would extend your arm. Now that's not going to happen in a general weightlifting scenario. Okay, the only time really that's going to happen is in a situation where the body feels like it needs to save itself, and typically that's going to be in a falling sensation. So if you've ever fallen, you'll notice that you usually throw your hands out and you break your fall automatically without even thinking about it with your hands to stop you smashing your front teeth out. So a quick bend at the biceps at the elbow, with of course the sensation of falling, switches off the biceps so they don't contract and turns on the antagonist to push back. So when you fall, outstretched arm, you push back and you break your fall effectively. So a classic example of a GTO in action is failing in the gym. You're really pushing through that rep and the tension going through the tendon, in this case the pec tendon, is far too much, and what happens unfortunately is the pecs turn off, and when the big muscle that's driving this lift turn off, game over. We're going to drop that weight. Now, people who are more likely to tear a tendon are definitely those who are seasoned lifters. They've habituated, again, they've changed their Golgi tendon organ to expect more stress, they've thickened their tendons, and they've voluntarily made their tendons accept more stress over time in a progressive type program. Whereas a newbie, if they tried to lift too much weight, they'd find they fail very quickly because their tendons are not strong enough and their GTOs are not developed enough to allow stress to go through the tendon, so their body will switch off the muscle very quickly, causing a weakness and a failure to occur much more often in unseasoned lifters. So there we go. We've reached the end of the presentation side of things. We've just got to dip back into our mock questions one more time and answer these final things. Okay, so here we are, question 15. You've got 3 options here and 3 multi-choice answers. It's very similar to what you can expect in the multi-choice exam. So, what have we got? We've got A, these send information to the CNS about the position and amount of movement of joints during movement. That is, of course, the joint kinesthetic receptor. Answer is A. If activated, these will inhibit contraction of the contracting muscle or agonist, and they might initiate action of the antagonist muscle. That is the Golgi tendon organ. And that leaves these that monitor the length of muscles, and if a muscle is overstretched, they cause contraction of that muscle. That is the muscle spindle. So C. So chucking those ones in. Alright, so quick reflection here. We've covered all of these aspects here. We've covered all of these questions here. Should be filled in as well. And we've covered all of these bad boys here. So the only thing that should not be filled in at the moment, I guess, if we haven't got round to it, is this little story here. I did mention what I wanted you to do there. If you haven't come up with a good little story yet showing your awareness of what goes on in these this sort of 3-sequence situation, then make sure you contact me and we can help you through that. You'll see that below we have the muscles question. That's the next topic we move on to. Thanks so much for listening, and we'll catch you in the next one. Cheers, bye.